improvement of resistance to rice blast by CRISPR-Cas9 mediated mutagenesis of PI21 gene in rice, that is Oryza sativa.
So the introduction
goes like rice blast is one of the earliest known plant disease. It is also known as rotten neck or rice fever because of the damage it causes to the whole plant and in different plant parts.
As such,
it has been reported in rice growing countries all across the globe. At first in India it was reported in 1989, and the expected grain loss from the disease can go up to 80%, like at max.
So about the
disease and the damage. So as I said, it is called as rice rotten neck because it affects so many plant parts, and it is a fungal disease. The pathogen is fungus, that is Magna porthoraisi, and it is one of the most damaging disease of all the fungal diseases known.
It is not just rice
that it is affecting. There are so many host organisms or the plant species it causes damage to.
Beet finger millet,
or like in finger millet it can cause up to 50% grain loss. In pearl millet it has been quoted to cause significant yield loss. Again in foxtail millet the yield loss can go up to 30 to 40%.
So this is just
a gist of how the fungus comes in, and because of the conidium
it causes the infection,
and it goes inside the plant parts. The episorium and the penetration peg followed by the invasive hypha that goes inside the spike or the neck or the awns of the plant. So the initial infections can go with the leaf lesions, which is helpful in phenotypic screening also. The physiological effect could be the reduced photosynthetic capacity, decrease in the plant vigor, stunted growth, poor grain development, and the reduced tillering. Obviously, everything leading to the reduced grain yield.
Thank you. So, as my colleague mentioned about the infection, how Magniporthoraisi causes the disease in various crops. There are various types: finger blast also, neck blast also. So many researchers are working on various approaches. Like breeding is the main approach. Sorry, Bindu is coming. So Bindu take charge of his.
Good morning, everyone. I would like to, as we are working on PI21 gene, which is responsible for resistance. Capital PI is causing susceptibility, and small PI is conferring for resistance. In order to find this gene which is conferring for resistance, we have to first find where researchers have crossed Nippon bear, which is moderately susceptible, and Hatamoki, which is resistant. And they have advanced the fourth generation. They have found that there is a QTL on fourth chromosome which is responsible for resistance. Later on, after fine mapping, they have found that small PI21 is responsible for susceptibility. But the thing is, it is a recessive gene. So our ultimate aim is to transfer the resistant gene into the susceptible background. And this PI21 being a recessive gene. This small PI21 which is being the recessive gene, if we have to go for back crossing, it takes many years. Why? Because after every back cross we have to do selfing also in order to transfer it to the next, in order to do screening. Because in back cross generations we cannot screen because recessive gene won't be expressed in the F1 generation. So we have to go for selfing and then do screening, which is a tedious process, and also it takes many years. Because it is taking a lot of time, we can go for marker-assisted selection, where the marker can be used for selection even during the F1 stages. Even though we go for marker-assisted selection, for complete introgression, it takes around 7 years. So in order to reduce the time of introgression and also increasing the accuracy, during back crosses there is a negative point, that is there may be undesirable linkages in order to curb that, and also increase the time for transferring. We can go for gene editing technologies. And the gene editing technologies will be explained by my friends.
So here for the infection Magnoporta oryzae uses this specific allele in this specific gene. There is formation of one domain which is rich in leucine repeats rich domain. So through that they form their interaction with the plant host interaction. So we target this susceptible allele gene for gene knockout approach. This is the region selected for the target for knockout. This highlighted region with PAM region we selected, and we check on NAB cutter. We found that the ZRA1 restriction site is there, which cuts between third and fourth nucleotide after the GG region. So this is the good approach for us to knock out this gene. For checking, this is the only one region of ZRA1 cleavage in the selected region. We just check here we check on that target region. There is only one ZRA1 restriction site. Along with this selected gene region, upstream of 200 bases and downstream 200 bases. On that gene region we check ZRA1 restriction site. There is also one restriction site is there. After that, with this specific restriction site, we digest it on that NAB cutter, and we found there is a single restriction cut in between this region. So next, chop tool, we check our target where it cleaves, which is cleave on intronic or exonic region. There are highlighted regions are exonic regions, and what efficiently they cleave. So here we found that above 80% we found the efficiency in their cleavage. So after that we designed the sgRNA in crispy tool. Here we found lots of sgRNA through that specific sequence. We selected some regions sgRNA for further mutation. So here we also see this specific highlighted sgRNA clues at various exonic region initiator to terminator sequences. So after that, this is enlarged version of these sgRNA cleavage patterns, and off target also they showing some off target also we found that the efficiency that sgRNA provided is higher than off target. Here constructing of sgRNA will explain Yamini.
So it is a simplified version of the Cas9 guide RNA construct. Herein we can see that the first thing is HPD, that is hygromycin, which will be useful in the selective medium while screening of the mutants. So it will be present obviously in the T0, and then in the successive generations we have to eliminate it because it should be free from the exogenous DNA. Followed by Cas9, which is going to encode for the Cas9 protein, and then TUNS, which is a terminator, and U6 is the promoter for the guide RNA, and the guide RNA being for PI21, which is the susceptible gene. Yeah, thanks.
Further in this step, we have obtained this protocol from the nature protocol. We have the basic steps like callus pre-induction. We all know that. Wherein we'll be using the seed of mature seed and de-risking and sterilizing it. Further in we'll be using this scutellum part of it, incubating in the dark condition. After co-cultivation we'll be going for the hygromycin selection. So later, after the aggregation selection, we'll put in the media for regeneration and cultivation of these transformed plants. Herein we have used this MTO 10110 rice variety. After this to find the mutation happened, we have designed the caps marker wherein after analyzing the digestion analysis, we'll get to know with the gel picture that if there is any mutation happened, we get only the single band over there, and in the wide variety we'll get the double band with the caps marker.
So after this SGRNA efficiently works there, we digest this susceptible gene. PI21 get knockout, and we found the rice blast resistance on that specific crop. This is our whole team which work on it. Thank you for giving us a chance to present here, especially NIPP scientist staff to assist us and guide us aware those novel technology with all the basic to advanced level. I am very glad to share our experience with you. Thank you, sir. Thank you.
A very good presentation. I have just few queries, not questions, about the nature of the protein that we were talking about. You said the gene is recessive in nature. So can you explain the nature of the protein at the protein level? What is the difference between the resistance and the susceptible as far as the protein is concerned?
Actually, it is a domain, sir, through which the rice organism is entering into the leaf tissues. If at all capital PI is expressed, that motive is active, and then the leaf pathogen enters into the leaf. If we can curb that.
My query is, suppose the allele is in dominant form. So it is also encoding a protein, right? And
Yes, sir.
susceptible allele is also encoding a protein.
No, sir. Susceptible allele is not encoding a protein.
It is not encoding any protein.
Okay, susceptible allele. It is encoding, sir. It is
So
encoding, sir.
So what is the difference between these two proteins? Why the one is showing susceptibility and the other one is showing resistance?
The one which is showing susceptibility is having a dominant allele.
So I understood at the gene level. I just want to understand at the protein level. On what basis you think that you are going to get the resistance in edited plant?
That protein is not
If you just think this is not a question, this is just my query. And second thing, you showed a construct. So I didn't find any promoter for the Cas9 and then your selectable marker. So which type of promoter you think best for your experiment? So my suggestion would be, because you are all students, this type of project, you focus more on the biology side. First see the function, the gene action, and whether they have anybody has fully characterized or not. What is its mode of action? What do you want to do? What do you want to do as far as manipulation and editing is concerned? So it is easy to edit any gene. But what kind of edit and how do you select this gene? And you said this was a QTL. So how many genes are there in that region?
In that 7 to 8 genes are they located in some articles, sir. So one is majorly causing susceptible alleles.
PI21 is a location or the gene.
It is a gene, sir.
What type of protein is encoding?
Some arginine and leucine rich repeats like domain creating through that pathogen get interacts and start their mitogen activated
So anyway, it's a good presentation. This is just a kind of hint and suggestion. You always keep these things in mind whenever you design any project for such type of students. Thank you very much. Organizers for inviting me to say. I hope you have learned a lot of things, and we are eagerly waiting for our Dr. Firoz sir. Thank you so much. Thank you so much.
Okay. So thank you. So now we take this opportunity to thank our expert, Dr. Sowat, please. We also take this opportunity to facilitate our floor coordinators and resource which were left that day because the experiment was conducted from the IARI. अच्छा संध्या Okay, so now I invite Dr. Firoz, scientist in genetics division, and he'll be talking about how to celebrate the Cas9 and other part of the tDNA for genome edited plants.
Very good morning. Respected course directors, coordinators, and co-coordinators, and inter-students. Very good morning. First of all, I thank the organizers for giving me this opportunity to talk on something like how the edited genes will segregate and what could be the implications. It seems very easy. A gene is edited, so everything is done. But it's not the case. Handling of the edited plants very important. Now I work as a maize breeder and I teach principles at division of genetics. And while working in maize, since maize has a lot of mutants and many of the mutants are in the grains, so we can see the phenotype of the grains. So that becomes maize as a model organism to study genetics. Now we have lot of tools in physics, chemistry, geography to show it to the students how the solar system looks like. There is a sun and on and around there are different planets moving around. How the Earth looks like. But in the case of genetics, there has not been anything rather which we can show it even the students, even the PhD students, how segregation works. So background, we started working on developing some of the mutants in maize, and we have now come out with a kind of series of genetic stocks through which we can show how the segregation works. Now, I'm not working on gene editing. I'm a part of a gene editing project. Very beginning. But the basic principle will remain same. So here, what I'll show, whether it's a natural mutant or whether it's a mutant edited by CRISPR-Cas9 system, the behavior will remain same. So essentially, we will encounter different types of segregation, different types of phenomena, and that is what we'll try to explain. I hope I'll be able to make the students understand how we can behave. Now these are the list of genes edited in case of maize. So there are a lot of genes improving yield. They are all edited. All have been edited, and there are reports. Then there is improvement of nutritional quality, then enhancement of stress resistance, then creation of the male sterility, and most of the male sterility are basically in the nucleus. That is the genic male sterility system. And then we have lot on even haploid induction. So these are series of lists, and it holds true for different crops as well. So a large number of genes have already been edited, and some of the stocks are already ready. As far as the varietal development is concerned, of course, it's a different scenario. It is a matter of policy decisions. So those things are coming up gradually. But as far as science is concerned, a lot of genes in maize have been edited. Now, this is a very basic slide. We should understand that how an edited gene behaves. Now, these are wild type allele, or rather we call it as say dominant allele, whatever we want to see it. So this pink colored circle alleles. I hope allele we all know that is the alternative form of a gene. One is capital A, another is small a. Capital A gives resistance, small a gives susceptibility. Something like that. Once an editing works, there could be three possible phenomena. Only one of the allele is edited. The other allele remains unedited because it may work, it may not. So we call it as a monoallelic mutation. And then there could be another two situations where both the alleles are simultaneously edited at the same place, and both the alleles are essentially same. 4 base pair deletion in both the chromosomes, or rather both the alleles present on two different chromosomes. The another situation could be the two alleles are edited, but at two different locations. So therefore, they are not essentially same as far as the sequences are concerned. So we call it as biallelic mutations. Now, these are the T0 plants. So therefore, only in a case of this scenario and this scenario, we will get the phenotype of a particular gene if the function is a recessive function, because it's a recessive homozygote. In this particular case, this is a recessive allele which is created after editing. Phenotype will not come in T0 plant. It will only come. The edited version is a dominant allele. So only in a capital A, small a, capital A. So it all depends upon which gene we are editing, and by editing whether it becomes a dominant allele or susceptible allele. Depending upon scenario, the expression. So in many of the cases, we may not get the expression of a gene in the T0 generation itself. So obviously, as in case of transgenic as well, we'll go for selfing of T0. We'll come to T1. If this is the case, we'll call it as heterozygous, and these are the homozygous. Although both are homozygous, the difference is here both the alleles are sequence-wise same. Here both the alleles are mutated, but their sequence-wise it is different. Now, once we self it, so heterozygous will behave like 1:2:1. One will be wild type homozygous, the pink colored allele, and then this will be homozygous. So finally, we'll get the expression here. It is a recessive homozygote, or the recessive allele, and of course, this will remain with the heterozygous. So in T1, we have to select this kind of plant. If it is a monoallelic homozygous, it's a very straightforward. By selfing, the situation will remain be maintained. But if it is this scenario, alleles are mutated, but they are essentially different because of the sequence variation is different. And by homozygosity, all will express the character of a edited gene, but essentially it will still segregate in 1:2:1 as far as the sequence of the gene is concerned. For some trait, we'll get expression in T0. Genetic reason I've explained. in
while
all of the case,
we should get our expression in
but it may
T1.
not happen
But
always.
it may not happen always. There are other genetic phenomena which I'll explain in my
So
later
this is the
slide.
basic.
So this is the basic
This is clear to all of us.
This is
then
clear
it
to
will
all of
very
us.
easy
Then
to
it will be very
explain
easy to
all
explain
genetic phenomena
all
with the
genetic
natural
phenomena
allele.
with
So
the
I'll
natural
explain
allele. So
all
I'll
the
explain
phenomena in
all the
genetics
phenomena in
by a
genetics
natural allele,
by
which
a
is
natural
otherwise
allele
there,
which is
one is
otherwise
dominant
there.
allele,
One is dominant allele, one is recessive allele, or we call it as wild type allele, or the mutated allele.
the two terms for the
The two terms for the
students
students I'd like to mention, wild type and mutated. These are the two terms, and another two terms are dominant and recessive.
So wild type allele
So
is the
wild type allele
allele
is the allele which is present abundantly
or in the higher frequency
in the population which is existing today.
While the mutated
While
allele
the mutated allele are the rarest of the rare allele which is present in the population.
It is nothing to do with
It is nothing to do with dominance and
When
recessiveness.
we bring both
When we bring both the genes, wild type and the mutated allele together in a heterozygous situation,
whether in a heterozygous
in heterozygous situation, the allele which gets expressed, we call it as dominant allele. The allele which doesn't express get recessive allele. So a dominant allele could be a wild type allele, dominant allele could be a mutant allele.
Similar is the case with
the recessive allele as well. But in majority of the cases, we find a dominant allele is most of the cases is the wild type allele, recessive allele is most of the cases are mutant allele.
Okay, now coming to the
Okay,
next
now
situation.
coming to the next situation,
What do you mean by dominance?
what do you mean by dominance? We all know that, but through genetics,
stocks
we are expressing.
this
Now,
is a round
this is a round colored round
which is
seed,
very much evident.
which is
That's
very
how
much
the maize
evident.
grain
That's
looks
how the
like.
maize grain
And
looks
this is
like.
a crumbled
And this is
seed.
a crumbled
We call it
seed.
as shrunken
We call it as
seeds.
shrunken seeds. We all know
sweet corn.
Sweet corn
is
is
a
a
corn which is having
is a corn
higher
which is
sweetness
having
and the
higher
green
sweetness at the green
stage, that is
green stage, that is 20 to 25 days after pollination,
and
there is an enzyme called ADP glucose pyrophosphorylase. So the larger subunit is contributed by a gene called SH2.
When it is
recessive homozygous, it will give sweetness, but upon maturity, since sugar is more and starch is less, packing won't be there. Since the packing is less, the grains will crumble. So by phenotype, it will very easily we can differentiate a round type grains vis-a-vis the shrunken type grains. Once we make a cross, this is the F1 seed. Now students let us know where will you get the F1 seeds? Which plant? You have a male plant, you have a female plant. Male female in the sense both are in maize, both the plants are bisexual, but one plant will have to treat it as male because it is the pollen which will be contributed by that plant, and another plant we have to treat it as female because it is the egg which will be contributed by that plant for the creation of the next generation of F1 seed. Now where the F1 seed will be produced? This is the P1 plant, say female. This is the P2 plant, say male. Where will you get the F1 plants? Where means on which plant? Where means on which plant? Female plant, right? Female plant, right? Female plant will give the F1 seeds. Now here you see the grains are round, and it is as similar as this. So therefore we can conclude this roundness is dominant over this shrunkenness, because in the F1 which is otherwise heterozygous, it is only expressing character of this allele. So therefore it's a dominant trait, and the trait which is not expressing, it's a recessive trait. Similarly, we have another example. Again the round seed, and there is another gene called sugary. There are two genes which are used in a sweet corn breeding program. SH2 is a gene which is responsible for higher sweetness. Sugar is a gene which is responsible for less sweetness, but it gives a good fragrance, or rather some kind of flavor. So in US and Europe, it is the sugary gene which is used in a sweet corn. In Asia and Southeast Asia, we always like more meat. You know, whether it's a sweet, we always like more sweetness. That's the reason why the SH2 gene which is having more sweetness, such kind of sweet corn is popular in India and Southeast Asia. If you go to Europe and US, you will get the sugary one. Now here, the crumbling is much less. It is like, resins, the kishmish type of phenotype is there. It's not round. It's just having a lot of, I would say, some kind of features which are very much there in case of resins. It is there. So it is a recessive sugary one, dominant sugary one, homozygous. Again the same story with the F1 seed. This is the F1 seed. The F1 seed is the round one. So therefore, roundness is dominant over the sugary phenotype. So just by crossing, we get the F1 seed, we can get such kind of scenario. We all know that, but then there are different ways to explain. Now we know there are two mechanisms
independent assortment, law of segregation, law of independent assortment, even dealing with the CRISPR-edited gene scenario, these two laws will apply. So in the earlier slide, I showed that it is segregating in 1:2:1. Typically, it is again following Mendel's law of segregation. Now we'll see how another way 1:2:1 is. So it's a normal round seed. It is a P1. Parent 1 is a sugary one. Parent 2 is a sugary one because it is crumbled. It is a parent 2. The F1 seed is round because it's dominant trait. Now we are selfing the F1 plant. So the F1 seed was on the female plant. We harvest it in the field. We grow it in the field. The F1 plant comes up. Upon selfing, whatever seed is formed on F1 plant is what generation? F2 generations, right? The seeds formed on F1 plant is F2 generations. In F2 generations, now you see there are round sugary seeds. There are sugary seeds. It is segregating exactly number is 1. The number is given 322. We counted in this, and then sugary we got 103. It is exactly 3:1. So this is how. Since it is a crop-based trait, we can see very easily. But if it is a plant-based trait, that is a foliage, leaf, or stem, we have to grow it, and this kind of segregation you will see yourself, right? In the field. So that is how 3:1 will exactly form. Another thing is, since if you grow the F2, this is basically F2 population. The seeds formed on F1 plant, if it is a F2 seed, so it is essentially an F2 population, right? And in a cob, we get around 400 to 500 seeds if it is a hybrid one. So you will have in a cob F2 population having 400 to 500 seeds. Okay, so it segregates exactly at 3:1. Then again for the shrunken, it's a round shrunken seed. F1 is round, and in F2 again, you see there are normal grains and there are shrunken seeds. So it is again segregating 3:1. So like that, for each trait, such kind of segregation will be there. Now look at here. So these are all F2 seeds formed on an F1 plant. You have to understand this thing. If you are working on a scenario, hybrid specially, the particular generation of plant will always possess the next generations of seeds. If the plant is F2, the seed will be formed on that F2 plant. If the plant is F3, the seeds formed on F3 plant will be F4. This is the F1 plant. On which seed has formed, so therefore it is essentially an F2 plant. So here you see, of course, round versus shrunken, 3:1. Here you see color. Anthocyane is a very major trait in maize. We call it as blue maize. It's very, very popular in Mexico and other places. In villages, in hills, especially in northeast, we have lot of colored maize. Here you see the colored versus white is segregating in 3:1. So therefore that is also a possibility. Here you see there are grains which are yellow. There are grains which is orange. These orange colored grains have got very high level of amylose. Normal maize has got around 30% amylose. Here it is almost 60. And because of the change in the starch proportion, the texture gets changed. So 3 will be yellow, orange. This 1 is orange. This 1 is nothing but there is a gene called amylose extender. When it is in a homozygous condition, we get this kind of texture. So it is again segregating 3:1. This is called the waxy maize. It is again segregating 3:1. Now you can differentiate this kind of grain vis-a-vis this kind of grain, white and dull looking. So this is because here the amylopectin, the other amylose, here it is amylopectin which got changed. Normal maize has got 70% amylopectin. Here we have made it to 100%. Because of the change in amylopectin and amylose ratio, the grains look dull. Again, it is segregating, and waxiness is a recessive trait. So this white colored one, you will see is segregating in 3:1. One is waxy, rest of them are wild types. So there will be various examples we will have like 3:1 ratio. We are showing it on a cob because it is easy to explain. If it is a plant trait, we have to grow and see such kind of segregation. Another important thing is there will be no order of how the recessive trait will be there. It's all random. Like the arrangement of seed having the recessive trait will be completely random. And each row and two cobs can never be same as far as the arrangement of wild type and seeds are concerned. Right? Now, the next law. the next law, that is law of independent assortment. It will only come when we are editing two genes and they are not in a form of a cassette. In a cassette, we will have several genes which are closely linked. So the entire loci will behave as a one locus. In such case, it will behave as there won't be any crossing, because there won't be any crossing over. But if we are mutating two genes at two different locations, which is far off, then independent assortment will come. In such case, here you see it is white. So there is one trait. So there is one trait. And the gene is Y1 we call it as yellow one. When it is dominant, it is yellow. When it is recessive, it is white. It is nothing but a gene called phytosynthetase, which has been used in preparing golden rice in case of rice. They have taken it from maize. So when it is dominant, it will produce carotenoids. All beta carotene and everything. It is orange. This first step is mutated, so therefore there is no coloration, no carotenoids. There is also another variation. This cob looks glossy and this cob looks dull. Why it is dull? Because here amylopectin is 70%, here amylopectin is 100%. It is a waxy kernel. So waxy cob will always look dull. We call it in genetic term cloudy appearance. Because it looks cloudy. It looks matte. There are photographs. One we call it glossy, another earlier used to be a matte finished photograph. So it is like this. There are two genes. Y1 is present on chromosome 6, wax1 is present on chromosome 9. Since they are present on two different chromosomes, it will behave as independent. They will behave independently. Once we go to an F2C, now here you see there are four kind of grains. One is yellow normal. We call it normal. This is glossy. So yellow normal and then yellow waxy. White normal and white waxy. It is also put it with an arrow. So there are four types of phenotypes we'll get because there are two genes. And each trait is a combination of yellow normal, yellow waxy, and white normal and white waxy. Normal means this is essentially the glossy the normal way of maize grains. Once we look at the individual trait, yellow and white was segregating 3:1. Normal and waxy was also segregating 3:1. But when we combine both the traits, it is segregating in 9:3:3. Now, if we are editing two genes, this is the one which will have both the genes edited in F2. Please try to understand this. Only one gene edited. Other gene remains wild type. What is the next three? The other gene being mutated. First gene being wild. Both the genes are wild. When both the genes are edited. Therefore, if we really want to say, and we are what? F2 means what? T1, right? T0 is your F1, right? Similar to F1, which is heterozygous situation. So total is 16 plant. Out of 16, one plant will have double mutant situation. If we get only 10 seeds or 16 seeds, only one probability or less than one plant will get. So it essentially means we have to grow large number of populations, good number of plants having both the genes edited, right? So population size is very important thing, which I'll again explain with a different example. Only by growing 16 plants, if we are expecting one plant to be double mutant, it's a theoretical expectation. Practically, we may not even get even one. Even you grow 20 plants, various reasons, environmental errors, right? So, I hope this is clear. To all of us. Now, a very important thing. Segregation of gametes. Basically, what we get in F2 is nothing but the two gametes getting fertilized, capital A and small a in some proportion. First, let us understand this. Then I'll explain in the context of how it is very important. There are pollens which is in a haploid state, and we collected the pollen and with potassium iodide. Potassium iodide is a dye which will bind to amylose and makes whether it's a kernel or whether it's a purple colored one, right? So the white dye which is having only 70% or rather 30% amylose, 70% amylopectin means 30% amylose. 30% amylose means blue color. It's a wild type. Capital A, once the waxy gene, which is wild type, is now mutated or a small WX1, then the waxy 100% is amylopectin. So amylose is nearly zero. So the coloration will be drastically reduced. So here the gives some reddish color, right? So homozygous plant, we collected the pollen, all the pollen red because all the gamete which we get are small w. All the capital W X capital W. But also which is capital S small. Capital S small. Gamete, one is capital A, one is capital A, another is small a. 80% of the pollen are dark, 50% pollen are red. So here you can very clearly see being produced by the F1 plant in equal proportion. So Mendel's two laws. Whether it's a law of segregation, whether it's a law of independent assumption, one of the assumption is both the gametes being produced by the F1 plant should be in equal frequencies. You make a checkerboard. If it is not 50-50, one of the gametes die because of some lethality. You never get 3:1 or 9:3 ratio in this stuff. So therefore, produced by the F1 plant. Here F1 is your plant, say heterozygous, right? Heterozygous. One allele is being only edited. So and if the gametes are not produced by 50-50 because red die because of pollen grains die because of some lethality or the other. So we will have lesser proportion of red gamete, higher proportion of the blue gamete. So in F2 progenies, you will have more proportion of the wild type genotype rather than the mutant genotype. We call it as segregation distortion. If your segregation is distorted from your expected ratio, it is segregation distorted. Now, if your recessive allele has got some lethality, so instead of 1 out of 16 or 1 out of 4, say 3:1, lot of one die, therefore your frequency of getting edited plant will be much less. Again, we have to grow large number of essentially get the edited. Because we never know which gene has got the segregation. We have the segregation distortion. Very important. Right? There have been recent examples 2-3 days back, article on paper, because of paucity of time, they mentioned the edited plant, the pollen expression is there, causing segregation distortion, and it is not following Mendel's 3:1. So I hope it is clear that it is not having 50 production because of some lethality or any other reason, then we are not going to have exact 3:1. It will be causing segregation distortion, and therefore 3:1, one is our aspirated homozygous. The proportion of mutated homozygous is further less because many of the homozygous plant will die. Many of the gametes will die at the upright state. So it is very important as well. The gene we edit should not have any segregation distortion. If there has, then your segregation will be. Next question is sampling. Very important. Now this is a crop. We call it as row. In maize grains, maize crop, the number of rows are always even order. 10, 8, 12, 10, 12, because the spikelets are always paired. So if it is one spikelet, there will be two floret. So both the floret being fertilized, it will produce even number of rows. Now you see the round F2 seed, right? This is an F2 seed, right? For sugary cheese. Same cob which I shown earlier. We have counted how many are round, how many are sugary. Row number 1, 2, 3, 4, and row number 1 is 3:1. It's almost like 3:1. If you do a chi-square test, it will fit in. The second row gives 5:1. The third row gives 3.25:1. Fourth row gives 4:1. The whole year, entire kernel is 4.26:1. And we took 3 years. And segregation of normal versus sugary 3.101. So therefore, if you are growing less number of segregated population, you may not get exactly this one. These two are 3:1, 4:1, and 5:1. So, right? But then this is number of kernels. When we go into the whole year, and then further year, you see the ratio actual 3:1. We also have to understand. Real terms, you will never get terms. You will never get 3:1. I should not say very rare to get exactly. You will always get very near to 3:1. That's why we test all have gone through huge data. Iceberg test is a statistical test which will tell you whether the ratio is fitting into an expected ratio of 3:1 or 9:1. So therefore, number of samples which you are growing for your analysis is very important. Just 4 plants should be good enough to have 3:1. But here we got 6 took 3:1. Here we got 4:1. But when we got almost like 1800, the ratio is very rationalized. 3.10 is to 1. So understand this. Number of population in P1 generation should be good enough to get the expected ratio. Please stop me when you don't understand. Very happy to answer. Next is a complete linkage. This complete linkage will come when you so you are editing 2-3 very close by. As I mentioned, the entire packet will behave like one. Instead of say 2 genes segregating 9 to 3:1 as a 3:1 combined. Now first let us understand this example. We saw this round seed and I explained. And there are no pigments. Very clear, right? Now, there's a genetic scenario like this. It is a round kernel. It is very closely associated with anthocyanin 1. We call it as A1. When it is anthocyanin 1, it is dominant pigmentation in other parts as well. And the two genes are only present very close by 0.09 centimorph. Base pair it is 140. When your cassette comes, it will not be this kind of. But in genetic terms, even though they are naturally very closely related. So therefore, it is an F2 seed. We took the round seed. We took the seed and we feed it on the field, and we found wherever we got the seed, it's inherently giving the purple color. Wherever it is the shrunker, it is giving the green color. No anthocyanin pigment. Because why? Because when you take the recessive allele, small a1, recessive gene doesn't produce anthocyanin. While the recessive gene of phenotype or sweet. So whenever capital A1 will go along with it, whenever small a1 goes. In genetics term, we call it as a coupling phase. Coupling phase is one linkage. Both the dominant allele and the recessive allele. Repulsion phase is one dominant, one recessive and the other recessive. So here, and then when what we did, 100% of the cases, round seed gave the anthocyanin based seedling, the truncated seed gave green colored one, anthocyanin less. So therefore, we can conclude these genetic terms are completely linked. Essentially means there will be no problem. But in real terms, completely is a relative term. Lots of probability here which is some crossing over, and you get a recombinant. But with a small number of we have not found for genetic reason we grew almost 5 lakh grains, and none of the 5 lakh we got to the recombinant. Small population related to these we also know molecular markers and other perspectives are here. This is a phenotypic marker. We don't know which plant has got chemical response. Just by looking at the kernel, we know this either capital or small because just by looking at the beautiful phenotypic marker which can be used in the market selection, not necessarily Coming to the next slide. The last slide. Now let us see what happens now. Uh, uh, you see, this is the selfie plants. All grains are formed. These are the small places where grains they not get. These are the places where grains would not be formed. This we call it as aborted embryo. The embryo could not be developed. It got developed. Now, why it got aborted? There is a gene called AMAPL. This is a very famous gene nowadays, which is getting edited nowadays. Pleiotropy is again the same thing.
Example all as
discuss this. Now, okay, I'm avoiding this. Okay, these are some of the new genetic stuff we are developing. Now, this. So with this, I'd like to thank
But I think, sir, in Mizoram that three crop is there, and all the three crops size is good. The size is good compared to what we are normally observing. Multiple crop, they are small in size. But in that Mizoram, that crop size is also good. Thank you, sir. Thank you, sir.
Yeah, yeah, please. Yeah, yeah, please.
As far as penetrance is concerned, if the allele is in recessive form, do you call it the absence of the gene, or so you consider it absence of the gene? Penetrance, what you call penetrance is other than the local allowing that is not allowing our targeted gene to get expressed. So it's a repressor. That is what it is. We don't know what is the background. Now we only know when we put it in the background, then first in one background you may 100%, others you may not have 100%. That's what it is.
Then there is no question of incomplete penetrance, right? No, because penetrance is purely genetic. So, right. The variability, whatever is coming in case of incomplete penetrance, it is because of the other effects from other genetic elements. So theoretically, if I assume the plants are all homogeneous for all other genes, then it will be. So there is right. So there is some part of environmental effect involved. And see whatever incomplete penetrance we get, does it perpetuate with equal frequency? Means say 90% penetrance. Will it remain in subsequent generation also? So what we usually say that many a times say that in case of genome editing, since we are developing mutant, so there is a difference with transgenic approach. Because in case of transgenic approach, you got to develop multiple plants to take care of positional effect and to get a plant with high expression. So that happens. That is applicable to genome editing also. But then it is desirable to develop multiple number. This
is one thing, and then this is one thing, and then you don't know how much will be the sort of expression because of other
So
factors.
many a times
So many a times we teach to the student that one of the advantage of genome editing is you may not require to develop many plants since you are creating mutations, so few plants will be fine to get the phenotype. But yeah, so yeah, here also considering the other phenomenon of genetics, it is desirable to raise as many plants as possible. That's why.
and then how is
And
it,
then how do you correlate this
variable
variable
expressivity
expressivity
with epigenetics?
with epigenetics?
Okay, so it may be one of
So
the
it may be
molecular
one of the molecular
reasons for
reasons for
variable
variable expressivity.
yeah, and
Yeah,
right,
and yeah, right.
know it.
Know it.
So there could be other
So there
phenomenon
could be
involved,
other phenomenon involved,
but these are all
but
underlying
these are all
reasons.
underlying reasons.
And then in case of
And then
doses
in case of
does it
doses effect,
applies
does it
only
applies
only when we talk
only when we
beyond
talk
diploid,
beyond diploid,
triploid onwards?
triploid onwards?
Few genes,
Definitely few genes, but
talk
we
about this
talk
phenomenon
about
only
this
beyond
phenomenon
diploid.
only beyond diploid.
Yeah. In case of
Yeah.
diploid,
In
the
case
same
of diploid, the same phenomenon would be called as
incomplete
incomplete dominance.
diploid.
Diploid,
Is it dosage
is it
effect
dosage effect
comparable
is
with
comparable with
dominance?
incomplete dominance?
Different. Yeah.
Different.
Because
Yeah,
biochemically,
because biochemically,
less
it is
production.
less production.
Biochemically, it reverses.
Biochemically it reverses. Those are very
Yeah. Yeah. Thank you, Firoj. I mean,
Thank
it
you,
was
Firoj.
so
I mean,
refreshing.
it was so refreshing,
So refreshing to
so
again
refreshing to
listen
again
about classical
listen about
genetics
classical
and from
genetics
such a
and
wonderful
from
teacher.
such a
You know,
wonderful
he
teacher.
was
You know, he
known
was
in,
known
at ARI
at
as
ARI
one
as
of the best
one
teacher,
of the
finest
best
teacher.
teacher, finest teacher.
So we are very
So
fortunate
we are very fortunate
that we could
that
have him
we could
because
have
he
him
has his own
because he
schedule
has
many
his own
times.
schedule.
He
Many
will be at
times
Hyderabad
he will
to
be
take
at
care of his
Hyderabad to take
experiments.
care of his
We are
experiments.
very fortunate that
We are very fortunate that we could have him within this schedule
of the training.
of the
Thank you,
training.
Firoj. Thank you.
Thank you,
Thank
Firoz.
you.
Thank you. Thank you.
Any
Great.
question from the
Any
online
question from the
participants?
online participants?
If there is any, please unmute and ask your question. Ask your question. Yeah, just a minute. Yeah, just a minute. Yeah, yeah. Firoz, then you come. Yeah, please go ahead. Yeah, please go ahead. So if there is no question from other participants, we may like to felicitate our speaker. For that, I request Dr. Bhattacharya that please come and felicitate Dr. Firoz. थोड़ा इधर आ जाइए Thank you. Thank you.
15 minutes. 15 minutes. 15 minutes.